Great. So my name is Enke. I'm at James Madison University in the Department of Biology for undergraduate research institution. I'll also double down on thanking Catherine for inviting me. This is a collaborative project. So my project collaborator, Chris Bernson, I think he's also here. He's really the sort of the structural biologist. I'm more of on the molecular side. And my our undergraduate student, Michael Friedman, has done pretty much all of the work I'm going to show with Chris. So I'm a little bit of a SAXS newbie, so please excuse me for that. The project I'll talk to you about is structural analysis of the photoreceptor-specific cone-rod homeobox transcription factor. So cone-rod homeobox, or CRX, it's a homeodomain transcription factor. It's sort of the master regulator of photoreceptor neurons in the human eye, as well as other mammalian retinal neurons. So photoreceptors are the rods and cones that absorb photons of light, transmit that light signal into an electrochemical signal, where it's transmitted into what we perceive as the visual world. So photoreceptors have tons and tons of highly specialized gene expression networks. CRX is sort of regulating all of those networks. Here down below, I'm showing a map from a previous publication of the CRX protein. So it has a DNA binding domain, a transactivating domain, and it's extremely clinically relevant. All of these mutant alleles that I'm showing are examples of human mutant alleles that result in loss of vision or complete blindness. And it's also striking that this protein has been studied for decades, but there's really no structural data beyond homology modeling. That's pretty much limited to this homeodomain, the conserved homeodomain. Outside of that, there's really no structural data for the rest of the protein. So that's really what we want to just try and understand. We want to get a basic structure for CRX to try and refine this model. So the process here is expressing and purifying the human CRX protein, sending it to the advanced light source, and generating some SAXS data, which we have in hand now, and then using this data to do some model-free and model-directed analysis to try and get an improved structure. So on the sort of biochemistry side of things, my student Michael has really become an expert at expressing and purifying proteins with the help of Chris Bernson. We're able to induce human CRX full-length protein in E. coli, fairly straightforward. We have a His tag on there, so we're using nickel affinity purification, in which we're able to get it fairly clean, and then we can further purify it using some size exclusion. So at this point, this is what we're sending off to get beamed. And some of our preliminary SAXS data that we've gotten back, the log I versus Q, P of R plots, these data are indicating that we have good stability. So CRX isn't falling apart when we smash it with X-rays. It doesn't seem to be aggregating. And then it also looks like it's sort of a good globular shape. So we kind of feel like we have good data in hand going forward. So Chris and Michael have been, they started out using some model-free analysis using the GASBOR technique, sort of all the specs over here for resolution, estimated volume, molecular weight. And what we got out of this initial analysis is, I'll have to admit, is a pretty satisfying sort of preliminary structure that we're kind of happy with. We've got the DNA binding, well, really the homeodomain portion over here in green, and followed by this linker region, which is a little less ordered, and then the transactivating domain. So we're still actively working on refining this model-free analysis to see if we can tighten this up. But pretty happy with this so far. This is pretty, I think we got these data. We sent this off in February, got the data back a little later in the spring, and then sort of COVID-related layoff. And this is what we've been working on this summer. Chris and Michael have also applied some model-guided analysis using the Ensemble Optimization Method. This is allowing us to take a little bit of the homology model data. While it's not great, the homeodomain homology modeling is very good, so we're able to sort of add that in with the SAXS data to get a little bit more guidance on how to predict this structure. And our initial analysis here is we compared our data, which is in these plots is plotted green, and we compared it to denatured or unfolded proteins, which are plotted in the red lines. And we could see that we don't really get a good match for our data. We do get a good match to more compact globular proteins. So we think this is pointing us to, yes, we have a nice, properly folded globular protein. So again, just kind of like, we think we have good data in hand that we can go forward with. So we feel pretty good about this. We're excited. We're learning more about how to do these analyses. So what we're currently working on is refining these model-free and model-guided structures. We're going to submit another submission to get beamed. We've ordered a homeodomain peptide for just that small homeodomain. We think that'll help us if we get some structural data for that, some X-ray data for that. That will help us with our model-guided analysis. And we think our preliminary data is suggesting that we have a nice, stable folded protein that'll be perhaps we can get a crystal and get a crystal structure. We also are very much looking for input from the BioSAXS community. I am extremely new to this. Chris Burnson, who is much more well-versed in this, he's also interested in learning. You know, any kind of tips that you all can give us would be greatly appreciated. And I'll just end by citing our National Eye Institute grant. This would be impossible without Chris Burnson and a really talented undergraduate student, Michael Friedman. So thank you guys very much. I appreciate the opportunity.